I'm not going to give you too much introduction, but I do sort of think it's important to explain to you why we think we'll see a signal in imaging, and then to show you where we're at in the process. Imaging endpoints are incorporated into 3 trials that we have ongoing, which are still all relatively small, but involve at least a couple hundred patients in total, and only one of those is finished. So I'm going to give you a little bit of animal data, showing you why we think we can see important signals in this data, and then I'll show you where we are with the analysis of the one trial that's completed as justification, and then what we hope we'll see in the future. A CT, or computed tomography, it's just high-intensity X-ray images, nothing that fancy. But the opacity to X-rays is measured in something called Hounsfield units, and that's kind of an important scale because we can use it to define things that are normal and abnormal lung volume. So Hounsfield units range from -1,000, which is air, to 0, which is water, to 1,000, which is bone, essentially. And you can characterize CTs and CT abnormalities based entirely on their density and the volume at which they occur in a normal lung. And I'll show you some examples of that. It's often called structural imaging because you're just getting a picture of what the structure of the lung looks like. And then I'm going to show you a series of studies using PET scanning, positron emission tomography. PET involves radioactive imaging of a probe. Typically in PET studies in oncology, the probe is 2-fluorodeoxyglucose, shown here. It's a really non-specific probe. It measures areas of active inflammation. Things that are burning glucose quickly trap the probe within the cells because they phosphorylate it to use the glucose, and the glucose accumulates in cells that are burning lots of glucose because they're angry. So it's inflammatory response, right? It's mostly macrophages, neutrophils, things like that that light up. And it's called functional imaging because it really gives you a picture in the lung of what's happening in terms of the inflammatory response to infection in this case. So I'll show you both human and animal data. So to get the animal data, though, wasn't necessarily trivial. We had to build a PET CT scanner that would work for medium-sized animals we were interested in studying. This is the dual rings. So this is a Siemens PET scanner, and it's fused to a human brain CT scanner. And the animal, in this case, a cynomolgus macaque, is immobilized on a bed that moves on a little tray through both rings. Then you collect data at different positions within that. And the first one's just a big whole body scintillation counter, and then it does the X-ray, the high intensity X-rays on the CT. And then they're fused together because they're co-registered. They're fused together to give you a single image that gives you both structurally what's happening in the lung and then functionally what's happening in terms of the inflammation within the lung. So let's start out with just a little bit of data from where we originally started with this. Well, actually, that's not true. We originally started with humans, then worked backwards to the animals. But I'm going to show you it the way that we'll present it in the papers, as though this was a nice logical story as we went through. So we started with rabbits. And rabbits, these are New Zealand white bunnies. You see one here getting infected with an aerosol of M. tuberculosis. They go through a hyperinflammatory response to disease, and this is just animals that were sacrificed to count bacteria. And then they have a chronic extended phase of the disease, which is relatively stable up to as long as we can tolerate having them in the facility. And that's actually published information now, so I'm not going to give you very much detail about the rabbits, but I do want to use this to illustrate a few things before we get to the non-human primate and the human primate data. So the rabbit, of course, is scanned this way. The sections you're looking at are going to be slices through the rabbit that are a few millimeters in thickness. And typically, this is the way, I keep shooting Tawanda in the head with the green laser. I'm sorry, Tawanda. Typically, this is the way the sections look. So the white and gray image is the CT dimension, and then we'll put on the radioactivity, so where the FDG is taken up. And typically, that's associated with lesions, but sometimes you can see uptake in the heart if the rabbit's particularly excited, and sometimes you can see activity in the bones. And so you'll see a scan will look like this. This is a 3-dimensional reconstruction of an infected rabbit showing infection in the lungs and the lymph nodes in this case, and then residual tracer down in the bladder because they retain activity until they urinate. So this is a single animal, and I'm just using this to show you the power of doing this sort of serial imaging techniques with this. This is a single animal before he was infected, and this shows you the CT. This shows you the PET on this side, and then this is the fused image, so you can get a sense of where the images overlap with each other. Before infection, the lung parenchyma is largely clear. Almost all that density is nearly all air because lung parenchyma doesn't have a lot of structure embedded with it. By 3 weeks after he's given an aerosol infection of M. tuberculosis, you can start to see some slight thickening of the lung parenchyma and some slight uptake in the PET dimension. And you can see after 4 weeks, so this is just a week later, you start to develop discrete lesions throughout the lung. And those lesions are typically, but not always, hot, taking up FDG because they're sites of active inflammation as the rabbit develops disease. By 6 weeks, just 2 weeks later, you can see there are extensive lesions throughout the parenchyma of the animal on in both lungs, because you got a good aerosol dose of TB. And you can see those lesions many of them are hot. Again, not all of them are hot. So remember, rabbits go through this sort of hyper-inflammatory response, and then they control the infection. And we can actually visualize this. Same animal, you just looked at the scan at 6 weeks. Here's what happens 3 weeks later. Actually, a lot of the lesions in the lung parenchyma have resolved. And so there's a lot of clear areas in the lung, and some of them persist. And the ones that persist, in this case, tended to be hot, and they tend to go on at 14 weeks and at 20 weeks to develop progressive disease. But you can actually visualize this inflammatory response, engagement of the immune system, control of the disease, formation of the granulomas in the cavities that the rabbit is ultimately going to have persisting throughout the course of disease. So rabbits are great because they develop very human-like pathology. In particular, they develop these caseous necrotic lesions we're quite interested in, and also in cavities that we're quite interested in. And as they do that, those cavities and those lesions during the course of disease actually grow more and more dense. And so I remember I told you about the Hounsfield scale. So this is just taking lesions at different time points post-infection and showing what happens to the density of these lesions over time. And as the rabbit contains the infection, as caseous granulomas and cavities are formed, all the lesions, as a general rule, get more dense, get harder and more opaque to X-rays. And so you can actually see that, and you can see these beautiful lesions like this one that then we can come in and ask, okay, now if we treat it with drug X, Y, or Z, what actually happens? And again, this is published, so I don't want to get too much into the details of this, but I do want to point out that in the rabbit, we were able to explore quite a few time points by serial imaging. And this on the top is the CT data showing 4 weeks of treatment with either INH or rifampicin, 4 and 8 weeks. And then you see the sort of general trend in the animals of what happens with lesions over that time period. So in a control animal, those lesions are stable, and in the treated animals, they resolve slowly. And that's critical for understanding the images I'm going to show you, because the slow resolution is because the destruction that the disease causes in the lung actually takes a fair amount of time to reverse itself, and we'll see the same thing in people later on. In contrast, this is the maximum SUV, which is just a way of measuring, comparing the radioactivity in animals that are treated with a single week of INH or rifampicin. And you can already see that there's significant signal, even in 1 week, in animals that are on successful therapy with drugs. By 4 weeks and 8 weeks, just to give you the corresponding picture up here, the signals are dramatically decreased in the animals. So the PET changes precede the CT changes by at least a month, and oftentimes by more than a month. So now I want to switch to a system that's even a little bit more like humans, and this is the non-human primate. These cute little guys are common marmosets, and they were our model of choice for developing for TB drugs for a couple of reasons. Number one, and probably most important, they're small. So these are the size of laboratory rats. They're about 250 grams to, we've never seen one as big as a kilogram, I don't think. But they fall within this group of squirrel monkeys and our New World primates that are, as a laboratory animal, are widely used, easily accessible, and relatively inexpensive. We developed a model for these that I won't discuss in a lot of detail either. I'm just going to show you a few key results from it. The other important thing about marmosets, though, is about 80% of live births in the animals are twins, which, if you're a drug guy and chemist at heart, is fantastic because every animal has its own control experiment basically built in, right? And the most important thing from a chemist's perspective is you only need about 3 grams of a compound to do an actual efficacy study in these, which opens up the door for things we can actually put through the model dramatically compared to the cynomolgus macaques or the rabbits. We've infected these with different strains of tuberculosis. Again, I don't think I'll go into the details here because it's not so important. You can see the infection apparatus. They're quite simple. You can actually relatively easily adapt the same basic concept that you use for infecting mice, for infecting marmosets. And as it turns out, they're highly susceptible to different strains or lineages of Mycobacterium tuberculosis, whether these represent ancient strains, the way we think about them today, or sort of modern strains that are in circulation. And so here's just an example of that and the kind of experimental design we like. The colors here represent, so each thing is an individual animal. The colors represent twins, and so these are sibs, twin sibs. And we can ask how reproducible is the infection by looking at the reproduction rates in siblings. We can ask for strain-specific differences across two different strains and infection differences even at low dose. And I call these Lübeck in bird to sort of remind me to explain to you that the Lübeck dose is a sort of high dose and mimics what happened in Germany in the small town of Lübeck, where children were accidentally infected with M. tuberculosis in place of BCG, where the infection rate and the rate of progression to disease was incredibly high, and the mortality was incredibly high as well. And so that represents a sort of high dose infection. Now that was intradermal infection with M. tuberculosis, so we're not doing that. We're doing aerosol infection. But we contrast that then with what happened on the US Navy ship, the USS Byrd, which set sail with a single infected sailor who then subsequently exposed all of his crewmates to a very low dose aerosol infection over a long period of time. And so we wanted to try to simulate both ends of that spectrum, because the amount of organism you give makes a huge difference in the outcome that you see in human TB patients. What we saw, as I said, was that marmosets are very susceptible to tuberculosis. We didn't know that when we started this. It doesn't matter which strain you use, but which strain you use actually has very profound implications for what pathology and what distribution you get. So CDC 1551, which is a strain that was isolated that was responsible for lots of skin test conversion but relatively little active disease, has the least virulence, so it's consistent with the epidemiology. Africanum, which I won't show you a lot about, which is another TB complex member that's been associated epidemiologically with extrapulmonary disease, also causes extrapulmonary disease in the primates. And a recent isolate of the Beijing lineage from South Korea is the most virulent in this animal model. And again, I won't show you too much detail on that. So here's what happens with disease progression. These are the three individual animals scanned multiple times over a 2-month period. This top animal infected with a Beijing strain progresses very rapidly to develop extensive bilateral disease with lots of collapse even in one lung. With the CDC 1551, the progression is slower, but the pathology is more advanced. You should notice that this is about 40 days and this is about 60 days. So this progression is considerably slower. And then with Africanum, you get primarily extrapulmonary disease. So you can tune, if you want to look at the impact in extrapulmonary disease, you could do that with your drug model. If you want to look at the impact on cavitary disease or on rapidly progressive disease, you could do that with the model. The twin animals behave almost identically in the infection. So here's the Beijing lineage and two twin animals. Here's two twin animals. So they had almost identical numbers of actual lesions that were produced. Here's two twins infected with different strains, just showing at the same period of time that it really is strain-specific factors that make the difference in the disease outcome, not host-specific factors. I think I'll skip that. Okay, so what happens when you treat them with chemotherapy? So again, we're very into quantitative measurements from these, so I've just put the numbers in there for your reference across here. So this is the pre-infection healthy lung volume. It's about 19 milliliters in an uninfected marmoset. At the time we start therapy in this particular animal, he has about a 50% total loss of normal lung volume that's replaced by lesions. As we treat him with HRZE, so that's isoniazid, rifampicin, pyrazinamide, ethambutol, standard first-line chemotherapy. He responds very quickly, so by 14 days, you can see the total PET signal has decreased dramatically. By 28 days, it's gone, and by 46 days, you start to see recovery of the volume. So again, you see this lag, this PET change early, and then the CT change lags behind the PET change. Okay, so let's not summarize, let's move to the humans. So this is, to my mind, to my knowledge, this is the first TB patient who ever participated in one of our studies and laid down on the double ring of a PET CT scanner. So the first ring here is the scintillation counter, and behind it is the CT. Again, it's the same as the animals. The scans are collected as the bed moves through that, and then they're computer registered by hard registration, so you can overlay exactly the structural and the functional images that you observe in these. And of course, what we're trying to do, this slide should have been at the beginning, what we're trying to do is to replace the sort of phase 2 methodology that we use based on microbiology with something that's inherently more quantitative and richer. And so this is, again, why we moved towards these imaging studies. I don't know how that slide got there. I'm not going to tell you about the details of the trials. I'm just going to summarize the results and should focus on the imaging stuff, but it's at least important that you understand the imaging timeline for patients who are on this study. So this is a clinical trial in MDR tuberculosis patients, in patients who are either on standard therapy or on standard therapy plus metronidazole in this case. And then we'll see this image in more detail later on, but then they get a PET CT and a high resolution CT at study entry, at 2 months, at 6 months, at end of therapy, which is 2 years, and then 12 months later. So they're incredibly long studies, right, which is why I'm only going to really show you the final results from one of them today. It took about 5 years to get patients all the way through this because it's 3.5, 4 years on study, and it took time to recruit, a year to recruit the first study. To do the analysis of this, we tapped into a programming group that helped us write custom software by which we could take CT scans. This is an actual patient scan, just a 3D composite of that, and then strip off sequentially layers until we were left with essentially only the lungs to do the analysis on. Make that sound easy. It was relatively straightforward adaptations, but there were a lot of tricky bits to it. But when you get to this stage, you can ask what's inside those lungs. And this, again, this is a real patient scan. In this case, what you see is there's normal lung tissue, or apparently normal looking lung tissue that's mostly radio transparent. And then you've got vasculature, which are these sort of green lines which represent the vessels running through the lungs. And then you've got all these TB lesions. And again, that's not a made up picture, that's a real picture. So there's thousands in an average patient. There can be thousands and thousands of lesions. When the lungs are big, there's lots of stuff going on. And the problem with the computational analysis is that blood vessels are approximately the same density as the lesions are. So all this stuff tends towards a radio density of 0 or minus 50 to plus 200 or so. And so in order to really look at the volume of diseased lung, you've got to learn to subtract out the vasculature, which we did using a sort of neighbor joining algorithm by which we could rebuild the vascular tree in a patient lungs. You can tell this is a healthy lung in that case, and this lung has extensive disease from top to bottom, and so the vasculature is more broken. After you subtract those, you can do a lot of analysis on the actual lesions that are present with it. And so let me give you an example, and then I'll show you the aggregate results. So this is a patient who was a 33-year-old male, culture-confirmed MDR TB, but he was susceptible to rifabutin. And so he's got rifabutin, a fluoroquinolone, and then a couple of companion drugs. He's got a good regimen on board. At study entry, you can see, just vaguely, because this projector is not very good, but you can see the radio-dense material in his right lung. And by 6 months, he's had a good response to that. So even the radiologist will say that guy got a lot better at that point. But of course, his sputum culture and smear was consistent with that. His symptoms were consistent with that. He finished chemotherapy by full 2 years, and a year after he discontinued, he was still disease-free. So that's a cure. That's what a cure looks like. If you look at the radio density in his lungs, and now this is absolute volume in cubic millimeters on this axis, and this is displayed by the Hounsfield intensities from minus 1,000 to 1,000. You see this region right here is blown up because that's the region where his lesions occur. And at the time he entered the study, he had exactly 20 milliliters, 200 milliliters of abnormal lung. That decreased at 2 months, and then it decreased a lot further at 6 months. And so the disease got that much better. I can tell you exactly how much better, and we define that as percent change sometimes, or absolute change in volume. We haven't really figured out what the best metric for doing the analysis is, but I'll show you some of that. So this is as percent change, and this shows you 2 months, baseline to 2-month change in CT volume of percent change, and the patients here are color-coded according to how they did. So success means fully completed therapy and were disease-free afterwards. Probable success means they were disease-free, but we didn't have microbiologic confirmation of that. We just had a health exam visit. And then people who failed are in red. So people who either died or failed or reconverted microbiologically are in red. And so you can see CT volume change at 2 months doesn't really do a very good job of clustering the failures out of that. By 6 months, it does a great job of doing that. And so now all the failures are displayed here. And so you can see the confirmed failures. And then this patient, who I'm not going to talk about too much, but who had chronic compliance issues late in the study, so was technically didn't respond as well as the others and was lost to follow up eventually. And then you have all these patients who were successes. And so you could draw a threshold here, or you can draw an ROC curve, and you start to get the sense that at 6 months, the CT volume really does well cluster patients who are on successful drug regimens from those who aren't. Okay, so what about the PET scanning? So this shows you now 0 and 2 months, study entry in 2 months, and you can see immediately the extent of disease. It's mostly right lobe, middle, and lower lobe disease. And you can see all this aggravated tissue here, which is the site of all of his TB disease, and then his heart, because he was exceptionally nervous. This is, then that happens from time to time with patients, and you just have to learn to ignore the heart. He still has a heart, but he wasn't nervous this time. And what you can see is sort of, if you look at this sort of at a gross level, the bulk radioactivity decreased, and it decreased quite substantially over that period of time. But if you look at the specific points in that curve and you look for things that at their absolute maximum radioactivity, what you can see is there's actually part of the lung, and that corresponds to this lesion right here, that got hotter over that 2 months. And so there are regions that, there are really detailed changes that happen in the PET CT signal that I think are quite interesting and teaching us something about the response to chemotherapy. Richard showed this slide this morning, but let me walk you through this because this is another interesting subject for a different reason. So this is another study that I'm not going to talk about any details of. He was a 23-year-old male with chronic XDR tuberculosis who was enrolled in a study where we delayed patients for 2 months, some of the patients for 2 months, or we started treating them immediately with linezolid in combination with, added to whatever their failing drug regimen was. That's the only way to say it. He was diagnosed 10 years earlier, treated with first-line therapy and then a variety of second-line therapies, to which he failed completely. His DST showed basically pan-resistant organism with a questionable result to pyrazinamide sensitivity, likely resistant. At the time he was enrolled in the study, we call it -2 months, he was hospitalized and given his initial PET CT scan, and you can see the extensive disease in the left lung. He was 2+ smear, 28 days to positive culture. He sat in the hospital and ate bad food for 2 months, and then we scanned him again. And what we saw when we scanned him again was that, I still think this is mind-boggling at some level, was that actually the disease, far from being just a repeat of what we saw, which was what we were kind of thinking we were going to see going into it, the disease is actually incredibly dynamic. Even after a 10-year history of disease, he's developed a new lesion right next to the heart. These lesions, you can't appreciate too well with this resolution, but you can see they've sort of shuffled around in intensity. Some of the lesions, like this little dot here, if my handle stops shaking, have completely gone away, and others have aggravated in that period of time. To me, that's amazing. That says that TB, even chronic TB, is a dynamic process that's an entirely local battle that's fought within the lung on just a time scale that I never appreciated before. At that time point, he'd gone to 3+ smear and 15 days to positive, consistent with his worsening radiologic factors. And then he started taking linezolid, added on to his prophylactic isoniazid and levofloxacin, to which he was resistant. 6 months later, he's completely clear. He's smear and culture negative and went on to cure his disease with linezolid treatment. So how did the PET do in overall then, looking at the study I was showing you before? This is now, jumping back to the metronidazole study because we're still collecting data on the linezolid patients. This shows you the 2-month response in the PET change, and again, this is expressed as percent change, and then the 3 failures, and then the 1 non-compliant patient who appeared to be an initial success and then later went on went on. And the PET at 2 months was easily as good as the CT at 6 months in predicting long-term outcome. Remember, this is ultimate cure, relapse, failure a year after a therapy is discontinued. And it's why we're excited about PET as a potential endpoint, because we've basically got everybody correct who ended up failing on the trial. Now, it's a tiny number of patients, and you can criticize that, and that's absolutely fair, and it's just suggestive. But it's why I thought it was important to show you the animal data as well, because I think the animal data really suggests to me that we will see these early changes and that we'll be able to translate those into quantitative biomarkers that in the end do predict how patients are going to do. And just one last point on that to sort of beat that home. This is comparing now 3 expert readers' analysis of the CT changes in those patients, in patients who are on an active regimen versus an inactive regimen, defined here. And then these are the corresponding PET changes in those same patients on an active versus an inactive regimen. And so you can see that the PET, which is inherently very easy to quantify, right? You don't need an expert CT reader. You don't need to pay a radiologist. We do occasionally, but you don't need to actually get the results. That PET is actually more predictive at 2 months than an expert radiologist at 6 months in predicting how good patients are going to do. And finally, just to tease, to show you a little bit more about where this is going in the future. This is a patient in Stellenbosch in South Africa, where we're doing a joint study with Gerhard Wurzel and a comparative study of the responses in South Korea with those in South Africa. This is a fully drug-susceptible patient who's being treated with quadruple drug standard therapy at study entry and then at day 28. So now we've pushed the time up by a month, and we can still see very reproducible quantitative changes in the PET signal, even as early as 1 month. I wasn't brave enough, unfortunately, at the time to push it to 2 weeks, but I believe that in a future study we could probably push it to 2 weeks and start to see reproducible changes in PET that really give us an indication of how patients are going to do in the long run. Don't have any follow-up data on these guys yet. That's a 200-patient cohort that'll be done in another 3 years. So you can invite me back 3 years from now, Deb, to talk about it. So my conclusions for this, PET, I think, changes to me really revealed the dynamic nature of TB infections, both in the animals and in the humans, and it's on a time scale that I didn't appreciate was on a scale of months. CT changes do offer a nice quantitative alternative to sputum microbiology, and I didn't say that, but the algorithm we've developed for doing the computational analysis is something we're working to develop a web interface for, so anybody who's running a clinical trial and wants to do quantitative CT comparisons on it can use that software for free to quantify changes. The PET changes do, during chemotherapy, predict the outcome in all 3 species, and you can ignore the last one, that's just a pathogenesis thing. So I've talked about the animal studies quite a bit at the beginning of that. That's all led by Laura Vaya, who's here in the audience, sitting right over there, and you can ask her questions about the animals afterwards if you'd like to. This is my group in Bethesda. They're sort of arranged in priority order because the chemists are in front, and then the biologists, and then the, sorry. And I should also make sure I acknowledge my clinical team. These studies have been incredibly complicated and stressful to do. We do very intensive, small studies to try and learn as much as we can. I want to single out Myung-Sun Lee, who's somewhere on that list, who's the lead clinician who's done a lot of this, particularly with linazolid stuff. But I also want to acknowledge our radiology collaborators, our computational collaborators, Twigs, which means the world's greatest statistician, who's, if you have any questions about the analysis, is sitting right over there next to Laura, and our collaborators at Stellenbosch University and at UCT. And finally, I want to acknowledge the Gates Foundation, who funded the first study with metronidazole and all the original PET-CT data, without which we wouldn't probably have been able to do any of that work and really have gotten started on this. As I said, we started with the humans and worked back to the animals. And I think if it wasn't for their willingness to take a risk on this, well, we would have none of this data today. So thanks for your attention. Hope to have some questions.